Ruth won an Investigator Award from The Lancet, a Wellcome Trust Innovator Award, 2 Olympus Foundation Awards in an Alzheimer's Research Forum and the Manchester City Council. And in her spare time, Ruth writes a book on attitudes and treatment to women in certain developing countries. So I'd like you to welcome Ruth to talk about potential involvement of reactivated herpes simplex virus in Alzheimer's disease. Thanks, Ruth.
Thank you. Can you hear me? Am I audible? Oh, good.
Yes, we can, and we can see your slides.
So hello, Ruth. Thank you very much. Now you will hear an excellent talk in proper English.
Sorry, I missed that. What did you say?
Ruth, please start your talk about.
Oh, thank you. Okay. Well, thank you very much. And I'd like to thank the Magda Salaki and others for the invitation. I'm very glad to be talking to you. Just sorry I'm not there in person, as I would love to have seen, love to have met people, and I would love to have visited Thessaloniki a second time. Anyway, I'll start now. This isn't going to be as philosophical as Professor Korchin's talk, but it does bear out, I think, some of his points about phenotypic conversion and various types of injury converging onto a specific consequence. Oh, it's progressing, I don't know why. Sorry, I can't seem to move the slides on. I don't know what's happening. Could you just wait a moment, please? Not doing it. Oh, okay, thank you. Sorry, it's okay now, I think. Yeah, just going to talk very briefly about virus characteristics. Well, their aim is obviously to replicate, and they can do so only inside living cells, so they don't want to kill the cells or the person. It's advantageous not to kill them. And they can do this in the case of herpes viruses by becoming latent. When they're latent in that state, they don't synthesize proteins. And in the case of HSV1, there's a latency associated protein is the only transcript. But they can be reactivated by stress, immunosuppression, inflammation, and a number of other factors. Reactivation causes a productive infection, and that means direct viral damage and also inflammation. And it's likely that recurrent events cause cumulative damage. The effects, of course, vary from asymptomatic to fatal. In other words, infect doesn't necessarily mean affect. So people can sometimes get classed as controls who are infected, but they're asymptomatic, which is unfortunate because it rather messes up any calculations about effects. But anyway, the effects probably depend on host genes. Is an example of reactivating what happens. You can get about 25 to 40% of people who are infected with HSV1 get cold sores, show cold sores, herpes labialis, but the other 60 to 75% appear asymptomatic. Anyway, what's the evidence for viral presence in brain? Well, it's been detected in brain by PCR in 6 different studies. The first one was by my lab in 1991. And this was the first microbe to be shown definitively to be present in elderly human brains. Presence depends on the brain region and the age of the subject. I say the first microbe, in fact, now it's almost a menagerie of microbes thought to be present in brain. But we think HSV1 is particularly important for reasons I'll be presenting. The prevalence is very approximately 60 to 70%. It might be greater in elderly and AD brains. In the fact that they're similar suggests that they're equally susceptible to HSV1 infection, which rebuts the argument that's sometimes been leveled that people with Alzheimer's disease are more likely to get herpes infection. This doesn't seem to be the case. As far as young people go, their brains, the prevalence was about 10% or less. I say approximately because we didn't have sufficient numbers of people to do very adequate studies because we were extremely short of money always. Anyway, we obviously wanted to look into why there was this similarity of frequency prevalence in elderly normals and Alzheimer patients. And ApoE4 was at the time being considered to be a major susceptibility factor. So we decided to look for that. And we got a very, very nice and clear-cut answer. We found that there was a very strong association between HSV1 positive Alzheimer's disease brains, but very low ApoE4 frequency in HSV1 negative Alzheimer brains, or in elderly normals, both positive and negative for HSV1. Well, we were able to really show or indicate that association was actually, it was a causal association, but I haven't actually got time to go into that. But we could eliminate certain artifacts, which say I won't talk about now. But having found this association with HSV1, we decided we'd try to look at the effects of HSV1 infection of cells in culture. And this shows the sort of results we got. These are human glioblastoma cells, HSV1 infected, stained for beta amyloid. And this is the uninfected cell. And you can see the staining is very much greater. In other words, there was great increased production of amyloid in these infected cells. And the same applied to production of abnormally phosphorylated tau. These are stained for p-tau at one particular phosphorylation site, but in fact, we got similar results for many other phosphorylation sites as well. So that's the infected, and this is the uninfected, and you can see there's a great difference between the two. We then decided to do a much more speculative experiment, which we were extremely excited and delighted to find it actually worked, but I'll describe what it was before saying that. We decided to use in situ PCR, which is an extremely difficult method and took several years to set up actually, but it's very informative in that you look at sections of brain, and so you can get information about location of various things that you're looking for, in this case, HSV1. And this is what stands for HSV1 by in situ PCR. Here it's stained for amyloid plaques by thioflavin S, and here's the merge, which as you can see is exact, very, very exact merge. This was a very, very, say, exciting result. Below it, there's another section, completely different section, which we did the same HSV1 DNA in situ PCR, but we stained for beta amyloid by immunohistochemistry. And that again shows extreme co-localization. And what we found was, in the case of AD brains, about 70-something% of the viral DNA is present in the plaques, but only 24% in the aged normals. And almost all the plaques contained viral DNA. We then decided we'd try the antiviral acyclovir to see what happens if you treat infected cells which are producing this. And by the way, I should say that vero cells produce the same amyloid and tau on treatment with an infection with HSV1. So we want to see what the effect of acyclovir was, and we could quantify it in these cells very easily. So this shows the percentage of, this is HSV1 quantified HSV1 proteins with increasing acyclovir concentration. You can see it decreases considerably, but doesn't go down to zero. There's a very simple explanation for that, but I haven't got time to describe it here. But a similar thing happened with beta amyloid. It goes down when you treat with acyclovir with increasing concentrations, doesn't go down to zero. However, in the case of p-tau, that the abnormally phosphorylated tau, that does go down to zero at a relatively low concentration of acyclovir. So it showed in general that yes, acyclovir produces these hallmarks of Alzheimer's disease in cell cultures. So from all this work, we formulated what I call the HSV1 AD concept. We found HSV1 resides latently in brain of many elderly people who can reactivate probably repeatedly and then causes damage, which must be limited and localized because otherwise they'd be showing signs of herpes simplex and encephalitis, which obviously in general they don't. But despite being limited and localized, if it's cumulative because of repetitive reactivation, we suggested that it then leads to Alzheimer's. We think the virus reaches the brain probably in late middle age as the immune system declines. We suggested that because we found it in high frequency in brains from elderly people, but a very low frequency in brains of young people. And what we showed was there's a strong association, which we think is causal, between the presence of HSV1 DNA in brain and the carriage of ApoE type 4 allele. We don't know why, but there's probably more damage in the ApoE4 carriers, or perhaps less repair. We also found, I didn't talk about this, but this was very interesting. We found that ApoE4 is a risk factor for herpes labialis, cold sores, in other words, the damage in the peripheral nervous system. So that is really rather nice. Cold sores, herpes labialis, are usually caused by HSV1. And so here again, you've got the combination of the virus and the type 4 allele conferring damage. And then next, we found antivirals protect against HSV1 damage in cell cultures. And this work is supported by over 400 publications using diverse approaches, cell and molecular biological, epidemiology, biological, virological, and GWAS methods. So it wasn't just the work in my lab, although my lab was the only one working on this topic for 10 or 15 years, which is why I talk mainly about my work or my lab's work. Anyway, sorry, now far more people are working on it. And I'd like to mention 3 recent major studies, which I think are really quite important, very important. Firstly, the study, one of the studies in Taiwan, this was very good because it had a very large population, because nearly everybody there is insured by a particular type of insurance. So they had information health-wise about a very large number of people. And what they found was that HSV-infected people, people who were severely infected, who had cold sores, severe cold sores, had a greater risk than uninfected, seronegative people of developing dementia. And they also found that treatment of HSV1-infected people, these people with severe cold sores with antiviral drugs, greatly reduced the risk of developing dementia subsequently. And 7 other studies have found a similar protective effect, which is very, very intriguing, because why prior treatment helps is uncertain when the life in the body of antiviral drugs is so low, but this is a very interesting point. Anyway, the next paper, recurrent HSV1 infection induces hallmarks of neurodegeneration. This was done by the group in Rome, De Chiara et al. In HSV1 infected mice, repeated reactivation of the virus in brain induced by thermal stress causes all these types of damage: synaptic damage, cognitive deterioration, and accumulation of AD hallmarks such as beta amyloid, tau, hyperphosphorylation, neuroinflammation, and so on. So this was very nice exhibit of how reactivation does cause AD-like damage. The next paper was by Cairns et al. At Tufts University. They found that HSV1 infection of the human-induced neural stem cells, brain cell model, 3D model, causes amyloid and P accumulation, gliosis, neuroinflammation, and decreased functionality. As you can see here, this is stained for HSV, this is stained for A beta. These are uninfected, these are infected. And you can see the big difference between the two, a big production of amyloid. Similar results shown for tau, which I haven't really got time to show. So this also was an important paper. And the third one was it showed, sorry, I should say, it showed that infection alone cause these AD phenotypes. I don't know, sorry if I, anyway, I'm going to move on now to talking about infection and what effects that has, various peripheral infections or infection in the brain for that matter. It's been found or acknowledged recently that cumulative infections are found to increase the risk of AD or stroke dementia. I say stroke because in a lot of studies, dementia hasn't been characterized specifically from the records or in the records as either AD or any other type. So I'm using AD Dementia just as this there. Also, some vaccines have been found to reduce AD Dementia risk. For example, in a study that I did in collaboration with epidemiologists in Manchester, shingles we found causes just a slight increase in risk of dementia, but Zostavax vaccination, Zostavax is the vaccine used in the UK, reduces risk of dementia quite considerably. And this has been substantiated by 2 other studies in the States where they looked at Shingrix, which is the vaccine used in the States. Shingrix effects does the same thing. Way back in 2001, there was a paper by Vero et al. In Canada, which suggested that vaccination is protective against Alzheimer's disease. And we suggested then the reason for that was that reactivation of HSV1 in brain occurs when infection occurs. And what's happened here? Sorry, it seems to have gone back. I don't know why. Yes, and we suggested that vaccination reduces the risk of Alzheimer's disease because it reduces the amount of injury it reduces amount of infection, severity of infection and frequency, and therefore it reduces reactivation of HSV1 DNA in brain. So I'd like to go on to experiments that we've been doing to study that, because I think it's been borne out. This is a study I've been doing with Dana Cairns and David Kaplan. Oh, I'm sorry, it looks like it's jumping, I'm afraid. I don't know how to get back. Yes, this is the slide I wanted. We looked at the effect of reactivation. We looked at the effect of VZV on the 3D brain model of Cairns and Kaplan. And what we found was that VZV does not cause formation of amyloid or formation of tau. However, it does cause gliosis and other similar effects to HSV1, but quite different in not causing the main hallmarks. However, what we did find, and it was very exciting, was if you have quiescent HSV1 cells in this model, made quiescent by treating with acyclovir to stop viral DNA replication, the VZV infection causes very striking reactivation of the quiescent virus. Shown here, this is HSV1 plus VC valacyclovir, just quiescent, not producing anything. This one is treated with VZV, and this shows HSV1 here. This is the same with HSV1. This is the same with HSV1. A great increase in HSV1 infection after addition of VZV. This is the A beta result in the mock, the ones that are not infected with VZV, but they're quiescent, no A beta virtually, but adding VZV and making infected with VZV cause a great increase in infection. So here the graph showing the LAT effect, how high it is in the mock infected, and later on with in the presence of VZV, it goes down to 0. And this is a VZV infection marker, which I won't talk about now. So this was a very exciting result. We've since done, but haven't yet published, another, which we think equally exciting result, percussive brain injury. You can apply measurable blows to the 3D model. And we've done that. And what we find is a very different type of injury, I should stress. That also causes HSV1 reactivation. And this suggests to me that the virus is pivotal in the brain's response to damage. I mean, it's known anyway that the brain axonal damage reactivates HSV1, but here's a completely different type of damage from infection, and that causes a similar reactivation of HSV1 in this brain model. Say in life, axonal damage is known to reactivate HSV1. So we ask ourselves, what happens in brain after an injury? For example, infections causing neuroinflammation or impact TBI. Do brain injuries lead to HSV1 reactivation? One question. Second question, what are the consequences of HSV1 reactivation in brain? The answer to question one, in life, many infections and several other types of damage, such as axonal injury, immunosuppression, are known to cause reactivation of herpes viruses and axonal damage, as I said, with consequent inflammatory and direct viral damage. In some cases, this is known to cause reactivation in brain. For example, COVID has been shown to cause reactivation, particularly of HSV1 and even more so of varicella zoster virus. So it's quite, these are quite commonly known effects. Then what's the incidental evidence of HSV1 reactivation in brain in life? Well, HSV antibodies are present in the CSF AD patients and age-matched normals. We looked at CSF of both types. And this showed, when we did this work way back in 2005, that HSV1 replication and expression occurs in brain. The virus is not just sitting there in brain doing nothing. Then we did a study, which was actually some years earlier, but not by PCR, where we looked for virus in brain by in situ hybridization, not PCR. And we detected virus in brain of patients who had been immunosuppressed and who were seropositive, but we did not detect it in seronegative patients, nor in non-immunosuppressed patients. The reason we didn't detect it in the other brains, or some of the other brains, was obviously because the technique is relatively very insensitive compared to PCR. However, under conditions where the reactivation would occur very readily, immunosuppression and so on, we detected in the brain of those patients. Then another thing was HSV1 DNA was detected in CSF of far more people than would be expected. That was in a routine lab that routinely tested CSF from various other labs, checked for presence of viruses. And this suggested that the virus reactivation is much more frequent than perhaps one had been thought. As I said, infectious axonal injury, stress, immunosuppression, known to cause reactivation of herpes virus in brain with consequent inflammatory and direct viral damage. So what's the experimental evidence of HSV1 reactivation in brain and the link to dementia? Well, the group in Rome used hypothermia, as I mentioned, to induce reactivation of HSV1 in brain of infected mice. And recurrent reactivation caused cognitive defects and accumulation of AD hallmarks. So that was a nice demonstration of what happened if you recurrently reactivate HSV1 in brain, at least in mice. And epidemiological evidence has been shown over some years. High correlation between HSV1 reactivation as demonstrated by IgM seropositivity and risk of AD dementia, shown by Lettiner and Löfheim, and especially so in the case of ApoE4 carriers, more recent paper by Löfheim. So answer to question 2, what are the consequences of HSV1 reactivation in brain? Does the damage from HSV1 reactivation lead to AD dementia? Well, HSV1 infection of the brain model leads to AD phenotypes, but VZV and that also HHV6, which we've tried recently, do not cause that, not all the phenotypes. So HSV1 transition causing AD might be unique. We don't know yet. We're going to try other viruses and also bacteria. Consistently, no other virus has been implicated in AD dementia, nor has any been detected as present at high prevalence in elderly brains, whereas HSV1 has. And reactivated HSV1 increases AD risk, and repeated reactivation, as I said, of the virus in brain leads to AD characteristics. So I'm going to really could summarize by saying that two very different types of injury, infections and brain damage, in the model at least, leads to both cases to reactivation of viral DNA in brain. And I think this is probably what is happening in life, and what we think, and the reactivation then leads to the occurrence of various phenotypes of AD and eventually to the disease itself. And I'd just like to show this very recent paper. It's got a lot of publicity. Levine et al. They examined potential links using 2 data banks, therefore a huge number of people, between viral exposure and risk of neurodegenerative disease. And they found the largest association was between viral encephalitis, mainly viral encephalitis mainly caused by HSV1. The largest association being viral encephalitis exposure and Alzheimer's disease. So that was a very nice support for what we've been proposing, that a virus is responsible for a large number of cases, not all, but we think bacteria are probably involved in work as well, and there may be many other factors. I agree with Amos, is multifactorial disease. I think everybody acknowledges that. So, but what we're showing is what he mentioned, actually, phenotypic conversion, different types of injury leading to the same endpoint. So I'll finish by just showing a very small number of people who've worked in my lab over the years, small because there's been so little money and such problems in publishing, unfortunately, due to such opposition from many other people in the field, which is now lessening, I'm very glad to say. And all these collaborators who have provided not only, in some cases, materials, but also a great deal of information. And I'll stop there and thank you all for listening.
Thank you so much, Ruth, for the excellent talk. We have time for a short time for questions, and I'll take my prerogative to ask you. So, what is specific to Alzheimer's disease? Why do you associate, or what is the mechanism associated the herpes virus to Alzheimer rather than to Parkinson or whatever other disease?
Well, we haven't looked at other diseases, but the results of Levine do suggest that viruses could be implicated in those as well. So I think there probably is different receptors in different parts of the brain. But I really can't answer that adequately because really so little work, or almost zero work's been done, because for some reason, any ideas of viral involvement have been really opposed so much that there's been, I think as a result, there's been very little work done on that topic. Parkinson's disease in particular, I think, should be studied in that respect.
And the other question I have is this close relative of herpes simplex virus 1, which is EBV, Epstein-Barr virus, has recently been associated with multiple sclerosis. Do you see any parallel between these two?
Epstein-Barr virus, you mean? Well, yes, I wonder whether, I think the mechanism that's been suggested is really quite different from anything we're talking about here, but I think that it does support the idea that viruses are probably involved in other neurodegenerative diseases. And I think, as I say, there should be much more work on these. Sorry, I can't answer that question either really adequately. Insufficient work done.
Ruth, it's
Any
Chris
other
here.
questions? Yes, please. Have the microphone there.
Yeah, Ruth, it's Chris here. Sorry. Thank you for your wonderful presentation. Can I ask, what do you surmise with COVID? Do you think postulate about potential dementia diagnosis down the track due to COVID?
Yeah, I think it's unfortunately, it seems a probability, and I think the mechanism is we were going to try the effects in this 3D model of what COVID would be. We had technical problems with the spike protein. I just hope we'll be able to manage it. It's a bit difficult actually working on it because Dana and David don't have facilities for, protective facilities for that for that virus. But anyway, we hope to do it with the spike protein. I think what's happening is that is causing, like VZV and HHV6, it's quite probable that's causing reactivation of HSV1 in brain. And in fact, as you know, there have been a number of cases where patients have shown signs of HSV1 reactivation and also VZV reactivation.
Yes,
so
please.
that's.
Hello, thank you very much for your presentation. I would like to ask.
Can you introduce yourself?
What's happening?
We seem to have lost.
You hear me now?
Yes, we can. We can
Okay.
hear you. Sorry, go ahead.
Name is Botanaki Hariliana. I am a doctor of medicine, and I am doing a postgraduate program now in neurodegenerative disorders. I have heard about the microbial theory for the pathogenesis of Alzheimer's disease and the role of beta amyloid as an antimicrobial agent. What I would like to ask is, I don't know, maybe there's a difference between bacteria and viruses, so I don't know if it's two questions and not one, but have you detected any preference of the microbes or of HSV1, for example, in a specific location, for example, in the temporal lobe to be detected there? And can that be the reason why in Alzheimer's disease we have the amyloid deposition in specific areas?
Well, no, I haven't done any bacterial work. My group didn't do any bacterial work. We didn't have enough money to really do the viral work very well. But there has been work done by Judith McCloskey on Borrelia and Brian Balin on chlamydia pneumonia, and also quite a bit of work on recently on oral bacteria, all of which implicate these viruses in AD. I don't. I think they haven't done anything about, or very little about, localization. I think in all cases, it's been shown, though, in vitro, in cell experiments, that all these microorganisms cause formation of amyloid, which seems to be possibly at low concentrations to be protective. Presumably, as the concentration increases, as infection increases, the amount increases and becomes damaging. I'm sorry, not quite clear what your question was actually asking. Does that answer it?
Yes, I think I have my answer. Okay, maybe I wanted to ask if HSV1, for example, or if specific bacteria go in a specific part in the brain, for example, in the temporal lobe.
Yes, well, we looked, we didn't do a very thorough examination because we couldn't, but we looked in the temporal lobe and the frontal lobes, and we did find it there, but not in the parietal cortex. So I think it's well known that it does localize, HSV1 does localize in the case of HSV1 encephalitis. The bacterial ones, I don't know enough about them to say, but I think I imagine they would have looked at specific regions where it's known that bacteria go when there's a known infection.
Okay, thank you very much.